My name is Jessica Hopfield, and I am delighted to be your host this morning for our Women in Science program featuring Dr. Sofia Axelrod. I'm a proud Rockefeller alumna. I graduated from the university in 1991, and I studied across two labs, both headed by Nobel laureates, one the late Paul Greengard, and the other Torsten Wiesel, who some of you will know as a former president of the university. And while there were certainly fewer women students or faculty members in my era, both of these men were terrific mentors for women in their labs. And I think in hindsight, part of it was they were both at the time married to extremely successful and creative artists. And I think that gave them an appreciation for what strong and ambitious women liked to do. While I was a postdoc here, I made the decision, which at the time was somewhat radical, that I did not want to pursue a basic science career, and that I was interested in using science to create drugs and diagnostics. And so somewhat sacrilegiously, I left the security of this campus to go to Harvard Business School, where I got an MBA. And ever since then, I've done things that have straddled basic research and the business community, trying to bring new products to markets. And mostly these days, I sit on biotech company boards. My life brought me back to New York in 2019, and it has been an absolute joy to reconnect with the university in various ways. I am a member of the Women in Science Committee, and I sit on the steering committee for the Kellogg Women's Entrepreneurship Fund. Rockefeller is really an extraordinary place to pursue a scientific career. For those of you who are relatively new to the university has an extraordinary commitment to tenure-track faculty recruitment, and particularly women. During the pandemic, 4 faculty were recruited, 2 of them were women. And our women faculty here are among the greatest scientists in the world: Corey Bargman, Leslie Vossel, Titi DeLange, Elaine Fuchs, just to name a few. These women are truly superstars, and I'm sure several of you have heard them speak at various university events. These women attract the best and the brightest. The last incoming class of graduate students was over 50% women. And I'd like to think some of that is that they look ahead and can see these towering women as clear inspiration. All of you in this room have been instrumental in the sea change for what it means for women to pursue a scientific career at Rockefeller. So thank you for your commitment to women in science and helping to make science more equitable and inclusive for all. The newest initiative of Women in Science is the Kellogg Women's Entrepreneurship Fund, often referred to as WEF, which has been truly groundbreaking. Since its inception in 2021, there have been 5 rounds of grants. More than $1 million has been awarded. And Sofia, today our speaker, is one such beneficiary. Coming back to the university for me has been a wonderful full circle experience to get to help out in modest ways. And it's been very rewarding to be able to come back and help to mentor women, particularly those that are interested in taking their science out of the lab towards developing drugs, diagnostics, or devices. It's through the Kellogg WEF that I met today's speaker, Dr. Sofia Axelrod, and we've developed a wonderful friendship. So I'm going to say a few words about Sofia and then turn it over to her. Sofia studied in Germany at the Eberhard Karls University in Tübingen, and then at the Humboldt University in Berlin, where she got her Ph.D. in biology in 2012, and she's been at Rockefeller ever since. She is a research associate in the Lab of Genetics, which is headed by Nobel laureate Dr. Mike Young, and she studies the function and regulation of circadian rhythms in sleep, as well as aging. And it's a fascinating intersection, as you're here today, between biology and human behavior. And she will explain the secrets to a good night's sleep, but it may also be the basis for important new treatments for other clinical disorders. Now, I should mention that Sofia is really quite unusual. In addition to being a great scientist and a molecular biologist, she's an author, she's an inventor, and she's an entrepreneur. So I'm going to give a shameless plug for her excellent book, How Babies Sleep. If any of you are parents or have grandchildren, this has extremely invaluable tips to help your baby sleep. I tease her that her next book should be called How Husbands Sleep. I've been actually using her tools on my 60-year-old spouse. They work, but please don't tell them because it will ruin the experiment. So Sofia is a recent graduate of a Kellogg WEF grant for a therapeutic project, which you're going to hear about this morning. I should also note that she participated in Rockefeller's very successful recent program called Managing Science and Scientists. It's a workshop this past summer with faculty from leading business schools that was jointly sponsored by the WEF and the Dean's Office, and I think is a great example of providing women and men with the tools that they need to run labs and to progress their careers. So with that, we'll learn a lot from Sofia, so I will welcome her to the podium.
Thank you, Jessica. Hi, everybody. Can you hear me okay? Perfect. I'm so honored to be here today and share a little bit about my research with you. And thank you, Jessica, for the lovely introduction. I'm going to tell you about my work in the lab, which is about sleep and circadian rhythms. When we think about circadian rhythms, most of us think about sleep. After all, circadian rhythms regulate when we're supposed to sleep, and that's kind of where most people stop thinking about circadian rhythms. However, what we and others have discovered in the lab is that we don't have just circadian rhythms controlled by clocks in the brain. We actually have circadian rhythms in all kinds of organs in the body. In fact, we now think that every cell in the body has a clock. And these clocks run with a period of about 24 hours, controlling cellular function and physiology and processes such as energy homeostasis, endocrine hormones, stem cells, synaptic activity, or the immune system. How is the clock set? Well, it's set by light. And if I shine a light on you every hour in a dark room across day and night, I can shift your clock. I can shift you later if I shine a light on you late at night, or I can shift you earlier if I do this early in the morning. And that's actually why we experience jet lag. People used to think it's because of the different language or the different air. No, it's the circadian rhythm, and it's a different light exposure that slowly shifts your circadian rhythm. What I've always found fascinating about this curve is how similar it is to the one my mentor and Nobel Prize winner Mike Young discovered in fruit flies many years ago, speaking to the universality of circadian rhythms, because all life on Earth had to evolve to predict when the sun is going to rise and fall. On a molecular level, and that's what Mike won the Nobel Prize for, he discovered that there is a genetic basis for circadian rhythm. He discovered the clock genes, which run the clock in every cell in our body. These clock genes, their expression rises and falls across day and night, and they form a so-called transcription, translation, feedback loop, where they control their own expression, which takes about 24 hours, as well as the expression of downstream genes. Now, what genes are we talking about? How many genes? Well, it turns out that 50 to 70% of human genes are controlled by these clock genes. That means that basically anything you can think of has an amplitude and has an expression that can be higher or lower depending on the time of day. So circadian rhythms can be understood not just as a way to organize our sleep, but really to create temporal order to the molecular, cellular, and physiological functions of our body to optimize our health. The conservation between different animals, because we all have to adjust to being on planet Earth, is why we can study and why Mike discovered the clock genes in fruit flies. In fruit flies, it turns out that clocks exist in various organs, from the sensory system, the brain, the blood-brain barrier, the liver, and they control things like memory, sleep, redox cycles, metabolism, and even mating. And interestingly, as the flies age, these circadian rhythms erode. In humans, the system has been expanded even more during evolution. Virtually every organ and every cell, from lymph nodes, epithelial cells, fat, muscle, and of course the brain, has clocks. These control the heart, the lung, the liver, the kidneys, the immune system, and proliferate connective tissues. And when circadian clocks in these organs get disrupted, we develop sleep disorders, but also get heart attacks, stroke, obesity, autoimmune disorders and increased infections, and even cancer. I will tell you today about 3 projects that I've worked on during my tenure here at Rockefeller. And it goes from very basic science in fruit flies to much more applied projects, which I've been able to focus on more recently, thanks to the Callan Women in Entrepreneurship Fund and all of you. First, why do we actually sleep? I have a hunch that that's why most of you are here. And of course, we all sleep. It's always interesting, but from a scientific perspective, we actually don't really understand why we do it. And by that I mean, on a molecular and cellular level, we don't fully understand why do we need to do it. You actually die from sleep deprivation faster than from not eating. So it's very important, and we don't quite know why. We do all sleep. And how is sleep even defined? Well, there is a couple of so-called hallmarks of sleep that include physical quiescence, posture changes, a lowered response to stimuli, making up for lost sleep, being affected by sleep drugs and caffeine, sleep following circadian regulation, and also genes, having genes that are maybe universally present that control sleep. And as you can see here, humans, of course, sleep, but also birds, reptiles, fish. My little fruit fly exhibits all these hallmarks of sleep, but it goes even further down. And I was actually shocked when a paper came out a couple of years ago that even such a little hydra, which is a relative of jellyfish, sleeps. So we now believe sleep is actually a universal function of life. And in fruit flies, we measure sleep by putting them in these little glass cuvettes. They walk up and down, and an infrared beam measures their activity across day and night. And that can be translated into sleep. And you can see here that fruit flies actually sleep all night, but then they also take a little nap or siesta in the middle of the day. So sleep is a very hot topic in science. And there are a couple of main themes happening where researchers feel strongly that that is what sleep is. And so one of these theories is the motor theory, adaptive inactivity theory, which means that we need to sleep to not move, to conserve energy. Another one is the synaptic or firing homeostasis theory, and that's that we need to stop being awake so that our neurons can basically recalibrate. Memory consolidation, of course, is something that you've probably all heard of, but this is such a specialized function of sleep that most researchers do not believe that that's why we actually sleep, because you can live without memories, but you can't live without sleep. Clearing reactive oxygen species is more new to the scene, and it has been shown that you need to clear these toxic molecules not just from the brain, but also from the gut. And finally, a paradigm-shifting discovery in the last couple of years was the discovery of the so-called glymphatic system, which is a system that basically flushes out toxins from the brain by increasing the distance between the cells in the brain. And this washing machine only happens when we sleep, and it can get rid of toxic proteins such as Alzheimer A-beta. So it's clear to me and other researchers that sleep is not just targeting the brain, but that this is a whole body phenomenon and is not just for the brain, but for the whole body, and that there are brain-body interactions. And so in my research on sleep, I've actually focused on the organ that sits right at the intersection between the brain and the body, and that's the blood-brain barrier. The blood-brain barrier is buried in the capillaries in the brain, and the cells that form it are called endocytes, and they line these capillaries, and they have junctions that basically seal off the passage of molecules between the blood and the brain. Controlled substances include ions, nutrients, metabolites, hormones, toxins, drugs, even water. Everything is highly controlled to ensure that neurons in the brain can properly fire. In human pathology, blood-brain barriers get disrupted during aging, during neurodegenerative disorders by drugs of abuse. And of course, the flip side of that is that it's very hard to get drugs into the brain. Now, why does it make sense to study something as complex as the blood-brain barrier in fruit flies? Well, they need to solve the same problem. They need to seal off their brain just like we do, because otherwise, each time they eat a banana, the potassium would render them paralyzed. So even though they don't have a closed circulatory system and the blood-brain barrier is located on the outside and not on the inside of the brain, on a cellular and molecular level, the two blood-brain barriers between vertebrates and invertebrates are strikingly similar. And of course, the fruit fly is much easier to access and manipulate, especially in genetic ways, than higher organisms. So how do we measure the blood-brain barrier in flies? Well, we inject the dye into the fly, and this dye can penetrate into the brain and the eyes, which are part of the brain, only in accordance to the permeability of the barrier. And then we can look under a specialized confocal microscope at the eyes as a readout and as a window into the fly's brain to detect how permeable the blood-brain barrier was. If the eyes light up, the blood-brain barrier was leaky and dye was able to penetrate into the eyes. If the eyes are dark, that means that the blood-brain barrier was very tight. And so I set out to answer a few questions around sleep regulation and function, the relationship between sleep and the blood-brain barrier. Does sleep affect blood-brain barrier permeability? Does blood-brain barrier disruption conversely affect sleep? And what are the molecular mechanisms? First of all, how can we manipulate these things? People have developed tools to sleep-deprived flies in a variety of different ways. And the one that I will explain here is actually by activating certain neurons that promote arousal in the fly and in humans, and that's the dopaminergic system. By activating these dopaminergic neurons, you can see here that the fly becomes completely sleepless. In the red line here, this is how they sleep normally at night and during the day, and then we activate these neurons and they lose all sleep. And then we asked, what about their blood-brain barrier? It turns out that flies that had been sleep deprived just for one night exhibit a much more permeable blood-brain barrier. And then you might say, well, you're destroying something, this is not a physiological process, but then we can turn off these neurons and let them catch up on sleep. And when they do that, the blood-brain barrier immediately closes again, showing that this is not an irreversible process, but an adaptive attempt to do something, and that the blood-brain barrier is probably part of the regulation of sleep. We then tested this in all kinds of other sleep-deprived flies. There are genetic mutants that sleep less, and in all of these different mutants, which have been aptly named sleepless, wide awake, insomniac, the blood-brain barrier is always more permeable, showing that there is a more general relationship between blood-brain barrier permeability and sleep. In fact, sleep and blood-brain barrier permeability are strongly anti-correlated. Flies that sleep a lot have a very close blood-brain barrier, and flies that don't sleep well exhibit increases in blood-brain barrier permeability. Or in other words, show me your blood-brain barrier and I'll tell you how well you slept. Now, to round this out, we went a little bit deeper and we wanted to understand, how does this work? What about the blood-brain barrier changes during sleep? Or is this only a one-way street where you affect sleep and then the blood-brain barrier changes? We know quite a bit about how blood-brain barrier permeability is regulated. People have discovered GPCR, G protein coupled receptor pathway, that is active in these cells. And if you modify any of these genes in that pathway, these cells that are normally forming this highly homogeneous sheet become dysregulated, and holes appear in the blood-brain barrier, and the blood-brain barrier becomes leaky. So we did these genetic modifications to understand what happens if we directly target the blood-brain barrier and make it less tight and more permeable. Does that also affect sleep? And indeed, it does. These flies exhibit lower levels of sleep than normal sleeping flies and flies with a normal blood-brain barrier, showing that the blood-brain barrier itself is the source of the sleep regulation and that the sleep and blood-brain barrier are tightly linked. We also looked directly at this GPCR, which is called moody. And moody expression is much lower in all sleep mutants compared to wild-type flies. However, sleep deprivation immediately changes the expression of this blood-brain barrier controlling molecule, showing that this is a bona fide biomarker for sleep. So in summary, sleep affects the blood-brain barrier and vice versa. This moody GPCR acts in the blood-brain barrier to change the blood-brain barrier and sleep, and is a novel sleep biomarker. In the future, to me, the million-dollar question is, what is going on? What is going across the blood-brain barrier? It seems like it's opening and closing to get either something out or something in of the brain, and future work will hopefully answer that question. I'm also interested to understand how this system changes during pathologies and aging. Because of what I said about the drug penetration into the brain, pretty much from the start, it seemed like maybe time of day administration of drugs or changes in sleep could affect the permeability of the blood-brain barrier. So I tested that by injecting not the normal dye, but different drugs into the fly and checking the penetration of those drugs into the brain after mild sleep deprivation. And it turns out that both of these antibiotics that are used for meningitis, as well as this brain cancer drug and this anti-anxiety drug, increase their permeability, their penetration into the brain after sleep deprivation, which shows that this is in fact the case. However, for this to be actually useful for human research and maybe clinical applications, this whole premise would have to be true, namely that the blood-brain barrier changes across day and night or with sleep states. So I actually partnered with a scientist at Mount Sinai called Sam Horn, who studies the blood-brain barrier in mice. And we did basically the same exact experiment. We injected a dye into the mice and looked how much ends up in the brain, but we did that 12 hours apart. And as you can see here, in these different brain regions, the cortex, brainstem, diencephalon, and cerebellum, the penetration of the dye differs by time of day, showing that indeed this is a conserved mechanism. In the next segment, I'll tell you a little bit about aging and our studies relating to diet and the circadian clock. Aging, just like sleep, is mysterious and very interesting to all of us, I believe. And people have uncovered a bunch of molecular mechanisms that drive aging. It's not clear, they don't really form a coherent picture of aging, but it's true that cellular senescence occurs, stem cell exhaustion, mitochondrial dysfunction, and what I will focus on today, deregulated nutrient sensing. Aging interventions then target these processes. For example, epigenetic drugs, clearance of senescent cells, stem cell therapies. And something very easy, though, to do is dietary interventions or targeting the insulin and mTOR pathway. It's been known for a long time that eating less, not more, has beneficial effects on our health. In scientific terms, this is actually called dietary restriction. And it's been shown over the last 50 years that eating about 20% less calories prolongs your life. That's been shown across the evolutionary scale from worms to monkeys. As you can see here, these monkeys are the same age, but this one ate less than this one, and it looks a lot better. More recent research, and that's where circadian rhythms come in, dealt with the question, is this just about the amount of food, or is this also about when you eat? And what about fasting? Is the timing important, or is it just the calories? And it has been shown that this time-restricted feeding or daily fasting improves heart function in flies. In mouse, is actually required for these effects of dietary restriction. So eating less calories is not going to prolong your life if you eat those calories randomly throughout day and night. There has to be a period of fasting, and that period has to be circadian every day at the same time. In humans, there are many studies now that show that time-restricted eating improves, or intermittent fasting improves various metabolic and health markers. Now, we wanted to get a little bit at the mechanisms of this and ask the question, does time-restricted feeding prolong longevity, and why? And if so, why? Now, what is a good model organism to study aging? Even mice live 2 years. So if you're going to do a longevity experiment in a mouse and you're going to repeat it 3 times, that's 6 years of a Ph.D. Flies, on the other hand, only live 3 months. That's still a long time for one experiment, but that's doable. And because we believe the underlying mechanisms, everything I showed a few slides ago, are conserved in evolution, we might as well start with something as primitive as a fly. So we subjected them to this simple experiment. Instead of giving them access to food 24 hours, we gave them access to food only for 12 hours. And then we measured their longevity. And it turns out that this simple intervention has a profound effect on their longevity and extends their lifespan by 20 to 50%. The longer you do it, 10, 20, 30, 40, 50 days, the stronger the effect. But it's never too late to start. If you give the flies access to food all the time in the first half of their lives and only then put them on time-restricted feeding, you still get a lifespan extension. It doesn't matter when you do it. Should you eat early or late? It doesn't matter. You can completely invert this feeding cycle and still get the exact same effect. And we think that's because it's really about the circadian clock. You're using food as a so-called Zeitgeber, which means time giver in German, to help the body understand when to do what. However, when we use one of these genes that my mentor Mike discovered that knock out the circadian clock, and up here you see actually the activity rhythms over 2 days, 4 days, 6, 8 days, you see how beautifully aligned the peaks of activity are from day to day. In this period knockout fly, activity becomes completely random. These flies do not know anymore when to be active or not. They cannot partition their sleep anymore to night. It all becomes randomized. And it turns out that when we take such flies, period or timeless, another clock mutant, the effect of time-restricted feeding on longevity goes away. That means that you need a functioning circadian clock for this circadian feeding intervention to have a beneficial effect on your longevity. Why? What do we think is going on here? Well, when we are awake, your metabolic organs do specific biochemical reactions. They synthesize bile acid. They synthesize glycogen and cholesterol. The pancreas secretes insulin. Fat is being made. The muscle degrades glucose. However, when you fast and when you sleep, the opposite happens. By opposite, I mean the opposite biochemical reactions happen in your body. The liver degrades glycogen. The pancreas secretes glucagon. Fat is degraded, and the muscle switches from glycolytic metabolism to oxidative metabolism. So this is important to understand because these biochemical reactions take place in the same cells, and having them occur at the same time makes no sense and would be a huge waste of energy that would negatively affect your health. So it makes sense to partition these processes temporally and separate them. And when you don't have a functioning clock, you can't do that. And we believe that this time-restricted feeding helps in partitioning these two processes, increasing the amplitude of the difference of these reactions and thereby promoting your health. And now the next question is where do you need the clock? Do you need it in the liver, in the kidneys? Do you need it everywhere? And so we have the tools to actually knock out the circadian clock in a targeted way in all these different organs. And when we do that, it was really interesting to see that if you knock out the circadian clock in neurons, nothing happens. So these animals are actually, in terms of their sleep-wake cycle, arrhythmic, but they still have a functioning clock in their metabolic organs. And they still benefit from time-restricted feeding. Same with the liver. However, when you knock out the clock in the midgut or the fly kidneys, you lose the ability to benefit from time-restricted feeding, showing that it's the clock in these organs that is required for the effect. So in summary, what I've shown you is that this circadian feeding can prolong longevity by up to 50%, and the lifespan extension is dependent on the circadian clock in the fly midgut and the kidneys to promote longevity. In the future, we want to understand even further what does the circadian clock in these organs actually act on, which are the exact pathways and genes that need to be activated by the circadian feeding in the midgut and the kidney to promote longevity. And also, there are actually differences in sex. This works better in females than in males. Why? And what is the effect of different types of dietary composition? Finally, I'm going to switch gears and talk about something that I've been increasingly passionate about. I joined the lab being a totally basic scientist. I never wanted to do anything translational, and I just wanted to understand things. But chronobiology is really a unique field that goes from fruit fly research to applications to human health, basically in one fell swoop. And so I want to tell you about my projects in this area. And all of that started when I was pregnant. As a lifelong insomniac, I thought, well, I have just fixed my own sleep by being in the young lab and having osmotically absorbed all the information about sleep and chronobiology. And now this is going to happen, and I'm never going to sleep again. Necessity is the mother of all invention. So I applied everything I had to this problem. And I remembered that in the lab, when we don't want to wake up our little fruit flies, we always use red light. Why? Well, because we know how the light actually instructs and sets our circadian clock. We have a specific receptor in our eyes called melanopsin, which is in specialized cells in the retina. And this is a blue light receptor. It is actually activated by light of 480 nanometers. And this light is what tells our brain and the master clock in the brain to start synthesizing the period protein and start the day, start organizing the temporal sequence of all gene expression in the body. So if you want to do that, you need blue light, and you get that in the morning by going outside. However, at night, if you don't want to do that, and if you want to keep your baby sleeping, you need to make sure that there's no light. And red light is like no light for the system because the receptor has such a narrow spectral sensitivity. And that's why we use red light in the lab. So because of that, I knew what I had to do with my Leia when she was born. You guessed it. I had to put her under red light. So I got quite serious about that. I bought red light bulbs. I bought blackout shades. I taped the blackout shades to ensure that no light would ever come from outside. I installed these red light bulbs in every lamp, and when I had a second baby, I did the same. And when they got older and they were scared of the dark, they still slept with the red light, which is fine from a chronobiological perspective because the red light does not change our circadian rhythm, and they slept wonderfully, and I felt great. And this is not just anecdotal. It's been shown that kids are very sensitive to light, especially at night, and you can completely wipe out their melatonin if you expose them to light at night. And we think that's because their lenses and their eyes are so clear. I thought more about how can we understand human sleep and baby sleep in particular, which is just a special case of human sleep, of course. And I came up with this notion of the triangle of sleep, as I call it, where human sleep, I believe, is governed by 3 processes: circadian rhythms. Are you getting the right light at the right time? Is your physiology at night preparing you for sleep? Sleep pressure. Are you tired enough when you're supposed to be sleeping? Or are you trying to spend too much time in bed and then you wake up in the middle of the night and your sleep becomes fragmented because your body cannot actually sleep that much? Or conversely, are you not actually getting your sleep pressure down to 0 by sleep depriving yourself? The third aspect is less important for babies, but certainly very important for adults, which is psychology. Are you relaxed and free of worry when you're supposed to be sleeping? And so I put all this together and wrote a manual, mostly for myself, but then also for other friends, and the whole thing grew, and I ended up writing a book about it, as Jessica graciously mentioned. That book got translated into many languages because I really think there was a need in a sea of kind of conflicting and useless advice to give parents science-based tools to help their babies and their children sleep better. I also developed products to aid with this system, red light products, and also an app to give parents science-based advice. Now, I've been focused on another aspect of light. Science tells us that we need bright days and dark nights to entrain our circadian clock. However, what we get in modern society is dim days and lit nights. Because we spend our life indoors, we don't get the light that we need to entrain our circadian rhythms. And at night, electric lighting and screens keep us awake and give our circadian clock the signal, oh, maybe it's actually not nighttime, and this causes circadian confusion and circadian chaos. So at night, the problem can be solved by using different types of light, like I described. However, what about the daytime? What about the signals that we need, like actually right now, to tell our clock that, no, we shouldn't nod off. We should stay alert, and we should keep synthesizing, period, and keep activating all these genes that will keep all our cells and our tissues healthy. It turns out that since we were hunter-gatherers, our modern-day light diet has significantly changed on the scale of 1 to 2 orders of magnitude. So modern humans are getting between 100 and 1,000 times less light every day than we actually need. Why is that important? Well, sleep is one issue, but because of what I described and the profound effect of circadian rhythms on all systems in your body, circadian disruption is actually a problem that is clinically relevant and can be a biomarker for progression of cancer. Across all cancers, circadian rhythm disruption has been linked to improved survival. So for any patient, their level of circadian disruption can be predictive of their lifespan. So this is a big problem, this light deficiency. And the idea of bright light therapy, which of course would fix this, is not new. Thousands of patients underwent bright light therapy, and it's been shown that it's highly effective for depression, for sleep disorders, for Alzheimer's, for Parkinson's, for cancer-related fatigue, and pain. However, doctors cannot prescribe this, and this is still kind of in the realm of alternative medicine. Why? Well, for one, there is no FDA-approved product that went through the gamut of clinical trials. But secondly, how do you actually detect circadian disruption? How would you know that a patient really needs this versus some other aspect of their health? And so with the Callan Women in Entrepreneurship Fund, I'm trying to basically address both of these problems and build a therapeutic modality based on circadian rhythms and light entrainment using bright light therapy, and also a biomarker assay that could detect circadian rhythm disruption. And that's shown here. So the idea is that in certain types of patients, in particular cancer patients, the amplitude of their clock genes is very low or even random, just like in these period mutants that I've shown you earlier. And certain types of blood tests can now detect that. And we can use this research to develop something that could be used in the clinic to detect circadian disruption in certain types of patients, and then correct their circadian disruption and optimize their treatment with light therapy. Now, the big innovation here, which I've been working on with this grant, is that modern screen technologies, which we know are so bad for your sleep at night, can actually be turned into something beneficial for your health by boosting it and using it, turning it into a bright light therapy device. So that means you can use software to turn an iPad into a light box. And now that's a much more attractive kind of proposition for a patient in a hospital to just read the news or play a game on their iPad while getting this absolutely necessary light that will help them get better. And then after a course of treatment, we can again repeat the biomarker assay to hopefully detect the increase in the clock amplitude and the restoration of their molecular circadian rhythm. So I've been working on this. We already have a prototype, and we actually are hoping to start testing this in patients soon. And in closing, I just want to say that this institution has been really special to me personally, because I was able to study this fascinating research, do this fascinating research here, both in terms of the basic science and now all the way to applied science. And some of the people who have helped me do this are in the room here today, and I think that's pretty special. So I want to thank you, and I want to thank all the people here on the slide, my mentor, who has been an incredible supporter of all of this, former and current members of the lab, Tom Sakmar, Bruce Conway, Jeannie Farrell, the Women in Science and Women Entrepreneurship Fund, at my colleagues at Mount Sinai, Tom Brennan, and my funding sources. Thank you.